Hello everyone. Today we are going to learn about Lac Operon, or famously called as lactose operon. Now we all know lactose is what? Lactose is basically something that we find in milk. What is it? It's a sugar. And it is a complex sugar, meaning it's a disaccharide. It is made up of two molecules. One is glucose, another one is galactose. Okay. Now, always remember, glucose is the simplest carbohydrate available for uptake, which is actually utilized by over 99% of organisms. So if suppose we have two options, let's say we have an option of lactose and we have an option of glucose, our body will first start occupying and metabolizing glucose. So it is that important. Now, if suppose glucose is not available, that is when lactose utilization starts. And we are looking in a very small organism called as E. coli, a very model organism to that extent. We are going to look at lactose operon or Lac Operon in E. coli. E. coli is basically a bacteria. Okay. Now, it's such a bacteria which is pretty much useful and found everywhere around us. So let's see. Before we even begin and dive on Lac Operon, let's try to understand what is operon. Okay, what is an operon? Operon is a cluster of genes that serve a common function and are all controlled together. Not much what we can say information we can get from this. Rather, I would say, let's see what is Lac Operon. Lac operon is an operon or a group of genes with a single promoter that encode genes for transport and metabolism of lactose. Now try to understand. They are saying transport and metabolism of lactose. They have even mentioned group of genes. Meaning, what is a operon? Operon is a group of genes which is carrying out one task. Correct? So now let's see what's given over here. Group of genes, cluster of genes that serve a common function. Common function meaning a common task. Lactoceron is a cluster of genes that helps in metabolism of lactose. Now comes the term all controlled together. Controlled together. Let's see any other word we can get for it. Single promoter. Now, always remember in genes, okay, we have this specific parameter. Okay. Genes have one starting point called as promoter and they have an ending point called as terminator. Okay. So, promoter is the starting point of the gene and terminator is the ending point of the gene. Whatever genes are present in between these 2 points, they would be the one transcribed. So these are the genes that would actually help us in carrying out any task. Okay, so let's say there are group of genes over here, group of genes. Okay, so group of genes all having a single promoter, meaning single starting point. Yes, that encode for transport and metabolism of lactose. So this is how we understand the actual definition of operon and Lac Operon. Let's have a look at how Lac Operon actually is represented. Something like this. There is a regulatory gene, promoter, yes, single promoter. There is an operator and there is a cluster or a group of genes. So can you see there are 3 genes? These 3 genes are responsible for what? For transport and metabolism of lactose. Okay. Now, what are we looking for over here? We are looking for in this particular slide, what are these 3 genes actually named as, or what do they represent, and what are their functions? Okay. So these three genes are namely LAC-Z, beta-galactosidase, Lac-Y permease, Lac-A transacetylase. This is a gene. Now always remember the central dogma. DNA makes mRNA, mRNA makes protein. So this gene is nothing but DNA. DNA LAC-Z makes a protein called as beta-galactosidase. Ase, whenever there is any compound or term or protein ending with ase, it indicates enzyme. So LAC-Z is a DNA gene. It makes a particular protein enzyme called as beta-galactosidase. There are two functions of beta-galactosidase. One, it cleaves our lactose to the simpler units like galactose and glucose. And second one, it converts our lactose into a more active form called as allolactose. So LAC-Z is the DNA gene. It makes a protein enzyme called as beta-galactosidase. Beta-galactosidase has two important functions. First one, breaking down lactose sugar into simple monosaccharides like galactose and glucose. And second function is converting the lactose, excess lactose, into allolactose. This is an active form. We'll understand the role of allolactose in some minutes. The second type of gene in the group of genes is Lac-Y. Lac-Y codes for a protein called as permease. Again, ase meaning this is an enzyme. What does this enzyme do? This permease protein actually helps in uptake of lactose. Logical, right? We are looking for transport and metabolism of lactose. Metabolism is carried out by beta-galactosidase. Transport, that is bringing lactose inside the cell, is carried out by an enzyme called permease. And the final protein that we have is transacetylase from the gene Lac-A. This transacetylase is actually not that prominent in function, but it is said to be an assistant to beta-galactosidase. Okay, so we have three genes. All of these three genes are helping in transport and metabolism of a sugar called lactose. Because these are a cluster of genes together coding for transport and metabolism of a function and have been controlled by one promoter, we call this to be an operon. Okay, so please make sure you remember the genes, what is the logic, and do not just by heart what's the Lac Operon or what is a operon. Take an example, understand it thoroughly, and then move forward. Now, this particular operon has got two sections. One is the structural genes, that is the genes that will be coded into proteins, and these proteins will carry out enzyme, right? Lac-Z beta-galactosidase enzyme, Lac-Y permease, Lac-A transacetylase. So these are called as structural genes. Another parameter we have is regulatory sequence or regulatory genes. Regulate meaning to maintain, to support, to stop. So any kind of regulation of these genes will be carried out by these three parts: regulatory gene, promoter, operator. Okay, so let's try to understand what actually happens. Central dogma says that DNA, that is the genes, they'll be transcribed into mRNA and then translated into protein. This also brings me into the other parameter. I have uploaded a video on DNA replication, so please do give it a go. Okay, now let's look at this regulatory sequence. This regulatory gene is also called as the Lac-I gene. This gene will be transcribed into an mRNA. We call it repressor mRNA. Okay, it's basically a repressor gene, and we transcribe it into an mRNA. This mRNA then gets translated into a repressor protein. Now, what is a repressor protein and what is its role? It's very important. This repressor protein, let's say, is like a phone. Whenever you're sitting to study, okay, your phone is the major mode of distraction, let's say Instagram. Okay, I have no grudge against any social media platform. So let's say there's an XYZ application in your phone, which is not allowing you to study properly for a good time. It's distracting you. Correct? So repressor protein is that protein. Okay. So just imagine we have a repressor protein that is a social media website or your phone. What happens? This particular protein will be worked in two cases, in the presence of an inducer and in an absence of an inducer. Let's say inducer is a teacher. Okay. And repressor protein is your mobile phone. Okay, inducer does what? It activates or allows the transcription of gene. So let's see over here. Let's say there is absence of inducer in your particular class or classroom. You're allowed to bring the phone and the teacher is absent. So what happens? This repressor protein, that is your phone, will stop your study process, right? Because you'll be completely distracted. So the amount of time that you should be giving to study, that would be blocked out by your phone because you're using it. Now, in that particular hour of study, when you had to actually sit and study, you were using your phone. So now you couldn't concentrate, you couldn't revise your concept, you couldn't attempt the exam properly, you didn't score good, you didn't get a good grade, right? So what is happening? This is an inhibitory molecule. Inhibitory molecule means what? Something that will stop the functioning. Okay, so what is repressor protein stopping? Repressor protein is stopping the functioning of the gene transcription. So why is that happening? Let's understand. From this DNA, mRNA needs to be made, and mRNA say we are getting protein. So because this DNA needs to be transcribed into RNA, pay attention, because this DNA needs to be transcribed into RNA, we require a particular enzyme which will help us make that RNA. And that enzyme is RNA polymerase. Okay, this RNA polymerase attaches to the DNA and it's going to actually transcribe the codes into an mRNA. Okay, now how is this RNA polymerase obstructed? Using repressor protein. So when there is no inducer, who is the inducer over here? Inducer could be lactose, inducer could be allolactose. Now, why am I calling them as an inducer? Let's play a logical sense over here. Lac Z, Lac Y, Lac A, all are involved in uptake and metabolism of lactose. Let's say your cell does not receive any lactose. There is no lactose in the medium. You have not included lactose in the diet. There would be no lactose for uptake. No lactose for uptake, no metabolism of lactose. Meaning, when lactose is absent, when inducer is absent, there is no point of running the machinery. Who will be metabolized? What would be taken in? Nothing. So that time, your repressor protein will stop the functioning. It's as good as saying that there is a factory and in that factory, milk is converted to curd. Okay. But let's say one day, milk was not received into the factory, was not bought into the factory. Now, would it make sense to keep the machineries working when milk was absent? Would there be any generation of curd? No. That's why repressor protein is going to stop the machinery of Lac Operon when lactose is absent. because there is no point in running the machinery when lactose itself is not there. How will it do it? It can't go and bind to the promoter. Okay, that does not make sense. So what is one gap between the promoter and the structural gene? It is operator. Repressor protein goes and blocks the operator. RNA polymerase, which was initially going to convert this DNA gene into RNA, when it attaches to the promoter and tries to go ahead, there is a blockage. Because of this blockage, RNA polymerase is not able to move up and it's not able to make an mRNA for these genes. No mRNA, no transcription, no translation, no gene products. There would be complete switch off of the machinery. So what is happening when there is no milk? The milkman or the industry man is going to turn off the machinery because there is nothing to be produced then. Similarly, when there is no lactose or allolactose, repressor protein binds to the operator and stops the machinery of Lac Operon. There is a controversial, or we can say an opposite case to this. What is that case? Let's say that the medium in which your cell is growing, the diet that you're giving to your E. coli, has got lactose. Now, if lactose is there in the medium, there should be something to pick up that lactose. Okay. Something to metabolize the lactose. Meaning, when lactose is present, these genes should be transcribed and protein should be made. Correct? So, presence of lactose, the genes would be switched on. Absence of lactose, genes would be switched off. So if we want the genes to be transcribed, our polymerase should run over here. And for polymerase to run, operator should not be blocked. So what does our allolactose or lactose do? It comes and binds the repressor protein. Teacher has taken away your phone, confiscated your phone. Now all your attention is focused on studying and you get good grades. Similarly, repressor protein is blocked using lactose or allolactose. It becomes inactive. Now, in such a condition, nothing can go and bind the operator. So, RNA polymerase can smoothly flow, transcribe the RNA, translate them into 3 proteins, and carry on the transport and metabolism of lactose. Now, one important thing which is not there actually in your syllabus, but it's very important from life science point of view. One of my students a couple of years ago had asked me, ma'am, permease is an enzyme or it's a protein which allows the uptake of lactose. So from where is this allolactose or lactose coming if the gene has not been transcribed already? So for that, you need to know that lactose has a leaky channel on the cell. So if suppose lactose is present in the medium, small minuscule quantity of it keeps leaking inside the cell. Okay. Like let's say there is a balloon. Okay, balloon, or we can say, yeah, you can take up a balloon. Okay, if there is no water surrounding it, no matter if there's a small hole in the balloon also, water won't be entering because it's completely dry. There is no water around. But let's see that the balloon has been kept in water and the balloon has a small hole in it. So some amount of water will definitely leak into the balloon. Same way is over here. If the cell is not surrounded by lactose, there is no way that lactose will leak into the cell. But when cell is richly supplied with lactose, the surrounding lactose leaks into the cells. Little amount of lactose comes into the cell and activates your Lac Operon. This might be out of your syllabus, but it's always good to know, you know. So once your lactose is present, things start working, and voila, your operon becomes operational. So what are the two conditions? Two conditions are absence of lactose and presence of lactose. Absence of lactose meaning there would be no product formation, no gene transcription. Presence of lactose meaning there would be product formation, genes would be transcribed. So there's a sweet way to know it. Okay. When there is no lactose, your repressor is active and it binds the operator, meaning there would be no transcription of structural genes. This is how we remember. Okay. When there is lactose, your repressor would become non-operational. Okay. It won't go and bind to the operator, as a result of which your transcription of structural gene would be switched on. Okay. So if you see, presence or absence of lactose directly affects the transcription and translation of the gene products. And this is how we understand and remember Lac Operon and its functioning. I hope you have understood this segment. Go through this again at a 2x speed in form of a memorization or revision. And without looking onto your screen, make sure to copy it down. Okay, just try to memorize how things are working, how we need to present it, how it is represented, and try to write it down in your notebook. That would be the best way to check whether you have understood or not. With this, we stop over here. Signing off. Bye.